from Texas Tech University, where he is an associate professor. Matthew earned his bachelor degree at Southwestern University in Texas, and his PhD at the University of Notre Dame in Indiana. He then returned in 2014 to begin a position within the Department of Natural Resources Management at Texas Tech University, where he is now. And his research has focused on the ecology of environmental DNA from the very early days of this field, from 2012 or so. And its applications to understand the distribution and dispersal of species, particularly invasive species, from freshwater ecosystems mostly, but not only. Some of his papers are well known in the eDNA community and are highly cited. However, today he's not going to talk about aquatic systems. He's going to illustrate us on airborne eDNA, an area of expertise where he is a leader, and he's been working on for many years now. He will discuss its potential to study and manage terrestrial habitats. He is also an editor of Biological Invasions. Any other journals?
Management of Biological Invasions and Bioscience.
Okay. And guest editor of many others. He's also the moderator of the eDNA Blue Sky channel. And he's currently teaching a semester abroad in Seville, which is why he is here. And he's, by the way, seeking opportunities or potential collaborators to develop a summer internship for undergraduates from his program back in Texas, but in Seville. So if you're interested, I'll let him know. And please join me in giving a warm welcome to Dr. Matthew Burns.
Okay, gracias, Ana, for the invitation to be here and talk with you today. Aprendo español, pero I'm going to talk in English. I have more English words than I have Spanish words so far. But as Ana mentioned, I'm here from Texas Tech, or I'm currently teaching in Sevilla the spring semester here, and me encanta Sevilla y España, and I'm looking for excuses to come back as soon as possible. So I'm really excited to share my research with you today, but really I'm here to advertise and hopefully meet some people that will give me an excuse to come back and do more research here as a collaborator. And as Ana mentioned, I'm going to mostly today talk about airborne environmental DNA and less of my traditional background, my training, but I want to start by just tell you a little bit about myself to look for all the possibilities to collaborate. And so my training is actually as a freshwater ecologist. And in particular, at the University of Notre Dame, we were studying invasive species, plants and animals from someplace in the world that get introduced someplace new and cause environmental and economic problems. And based on where we were geographically, the system that we often looked at was the Mississippi River, which goes through the middle of the United States, the continental United States, and artificially connects to the Laurentian Great Lakes that sit in between the United States and Canada. And because of that artificial connection, it's a really important connection for the movement of aquatic invasive species, both from the Great Lakes, which is a really important commercial shipping pathway, into the continental United States, as well as from the continental United States, the southern United States, up into that Great Lakes ecosystem. And so what you see here are bighead and silver carp, sometimes referred to as Asian carp. These were introduced into the southern United States in the 1970s because they are very hungry fish and do a very good job of keeping other fish farms clean. And in particular, these fish would be stocked into catfish farms in the state of Arkansas in the southern United States and keep those catfish farms clean. Well, during some flooding in the 1970s, the fish escaped their farms, got into the Mississippi River, and started eating their way up the Mississippi River northward until coincidentally, when I was doing my PhD at Notre Dame, they were right at the back door of the Great Lakes, and there was a lot of management concern that they would get into the Great Lakes, and because of their voracious appetites, they would really destroy the food web and the ecosystem. You also see here another concern about these fish. So they've got this interesting anti-predator behavior that when they are scared, they launch themselves out of the water. They jump to safety. And so you can go on YouTube and see videos of people riding their speedboats down the river, and these fish are jumping and jumping. And this is a very large fish, 100 pounds. I'm sorry, I don't have the kilograms on that, but this is a very large fish. There's actually stories of people getting hit in the head by a fish and passing out and falling in the water and drowning and dying. These fish can cause injuries when they jump. So not only are they very dangerous to the ecosystem, they're very dangerous to people as well. The problem is, with studying this invasion, at the leading front of an invasion, where they're advancing, they're not at these high population densities. There's just a few fish, just a several, and they're hard to find. And so our team started to wonder, is there something, we can't catch them with nets or electrofishing, how else could we look for these fish in these habitats? And that's where we started to explore this idea of environmental DNA. Organisms passing through their environment shed genetic material in cells, in wastes. Freshwater fish urinate nearly constantly. And so there's a lot of material, genetic material going into the environment. And so without seeing the fish themselves, we could actually scoop up some water, filter the fish DNA, the material out of that water, and use techniques like PCR to look for genetic clues that the fish is present in the environment. And I didn't find a good analogy for this, but in the United States, we have a very popular show called CSI, or Crime Scene Investigation. And so our lab back then, and indeed my lab still today, operates as sort of a crime scene investigation environment lab. We're looking not for criminals, but for rare species in the environment. Invasive, newly introduced species, threatened or endangered species, or really anything that might be hard to trap, track, and find using traditional methods. And just to show that my main background is in aquatic ecology and freshwater ecology, I just want to show a list of some examples of collaborators and projects that I've used environmental DNA methods to study, primarily in the United States and primarily in my home state of Texas. So for example, these are zebra mussels from the Ponto-Caspian. I believe they have caused their fair share of trouble across continental Europe. They're a very nuisance species in North America as well. We can use environmental DNA to look for them at the early stages of their introduction when eradication might still be possible. In the middle here, here's one of our big-headed carps that we have looked for in the environment as an introduced species. This is a mussel. We have done work in the Pecos River in Texas and New Mexico, looking for threatened, endangered, and rare species in the environment. And here is one of my current PhD students, Sadie Roth. She uses environmental DNA in the Sonoran Desert out in the western United States to look for amphibians and amphibian pathogens to try and help their management. Okay, but so what I'm here to talk about today primarily is my emergence from the aquatic system into the collection and analysis of airborne environmental DNA. So no longer are we necessarily scooping up water, Everybody really quick, do me a favor and take a quick deep breath. Okay, now you've all sampled airborne eDNA as well, right? Anyone that suffers from allergies is very familiar with the fact that some species shed a lot of DNA into the air around us. And I had a PhD student come through my lab. He was a terrestrial plant ecologist by training. And he showed up in my lab and he said, well, could we use DNA methods to learn about terrestrial plant communities without having, it sure would be nice to not have to measure out a big transect and walk and identify the different plants. Maybe we could just collect some air, collect some dust, and learn about what lives on the landscape. And so I'm going to tell you about Mark's work, but I do want to point out that just to get to one of the punchlines right away, airborne environmental DNA analysis has really taken off. And here is a graph that student, Mark, and I included in a review paper. I think this was the first. Airborne eDNA is a developing enough field now that we've already been able to write a review paper on it. But we show here the number of airborne eDNA manuscripts over time, and we see it's really increasing exponentially. I've got this 2024 bar in blue, just to remind myself to point out to you that we wrote this paper in 2024, and so that bar was incomplete, and at the end of the year would have been up there. So airborne eDNA is taking off, and I'm going to tell you a little bit about the role that I and my team have played in its development and how we've gotten to where we are today. And so I want to cover three main themes today to explore our path. The first one, I'm going to tell you the story about how we learned to apply airborne eDNA to learn about terrestrial plant communities. Then I'm going to tell you about how we started to ask more ecological questions, not just can we do it, but what can we learn using airborne eDNA? And then at the end, I'll speculate a little bit about where we might go from here. And so Mark, when he came to me, he asked, could we do airborne environmental DNA analysis and learn about the terrestrial plant community? And I said, I have no idea. Let's find out. We have at Texas Tech, so here's that's not to scale, but we're up in we call that the panhandle of Texas. That's where my university is located. We have this experimental rangeland property. It's fairly untouched, it hasn't been grazed in decades, and so it's a fairly natural plot of land in an otherwise developed city. And what we did was, or what Mark did, was he set up dust collections, polvo, I think, collecting stations across the landscape in a gridded pattern. You see here, this is what the collector looks like. It is a little triangular piece of metal flashing. You see on the front, there's a hole that is open. And the idea is that the wind blows into that. This top of the pie slice is a mesh screen. And so the wind blows out the top, but any dust that it happens to be carrying is deposited in the bottom of that pie slice. And this thing is set up on a post in the middle with some fins in the back. And so when the wind blows, that pie slice is always facing into the wind. And so the wind always goes into the pie slice and deposits material inside. And so he set up one of these collectors at 9 points across the landscape. And he would go out and he would, after those being deployed for several weeks, he would go out and I'm an aquatic ecologist by training, so he said, how do I collect this DNA? And I said, well, let's take your dust and let's turn it into an aquatic sample because I know what to do with that, right? So he would go and he would rinse it out with distilled water, And then he would take that distilled water back to the lab, and we would treat it like an aquatic eDNA sample. He would filter it, and then we would use standard DNA extractions to get the DNA off of that filter. And then we would use either PCR in the first couple of experiments that I'll show you, and later sequencing to learn about the DNA that we caught on that filter. And all the sequencing that I'll do today, or that I'll show you today, I'll point out, was done at the Texas Tech Center for Biotechnology and Genomics. So the first thing that Mark found out in his experiments is that the methods that he chose influenced how much DNA he collected. This isn't too surprising. We've seen this in aquatic and terrestrial systems as well. But what I want to show you, so he used, I'll point out, he used a universal plant primer. So he was just amplifying plant DNA. We're not distinguishing between different species yet. But when we quantify the amount of plant DNA, the other thing I'll point out is these are cycle threshold values. In other words, in PCR, this is, you can imagine, this is how quickly we get enough DNA in our sample that it starts fluorescing and is detectable. And so lower numbers mean more DNA, and higher numbers mean less DNA. And Mark used 3 different DNA extraction methods to get DNA from his samples, and found that there was a significant difference, that different extraction methods yielded different amounts of DNA from the same filter, subdivided a couple of different ways. Now over here, we've got 2 different universal plant primers, one targeting ITS2, which is a nuclear DNA gene, one targeting TRNL, which is a DNA from chloroplasts. And we found a significant difference here as well. Targeting chloroplast DNA, we got more DNA in our samples than targeting nuclear DNA, and that makes sense because there's one nuclear genome in each plant cell. There are many chloroplast genomes in each plant cell. And this is similar to what we've seen in animal studies. In aquatic systems, for example, there's more mitochondrial DNA in the environment than there is nuclear DNA in the environment, because your cells have many mitochondria and one nucleus. But so the lesson here is that our methods matter. Nothing too surprising yet, because remember that slide with the I talked about seasonal allergies. Certain plants we know release a lot of DNA into the air at different times of the year. And so what I asked Mark was, are we just detecting those allergens? Are we just detecting pollen? Because everybody knows that we can do that. And if all we're going to collect is a bunch of ragweed, I don't think that's going to teach us a whole lot. And so what we did was we designed 2 species-specific assays. One targeted this blue grama. It's a very common grass on our rangeland property, and it's wind pollinated. So this is one that evolutionarily is adapted to release a lot of DNA into the wind, and we would expect its DNA, a lot of pollen, we would expect its DNA to be in our traps. This other species that we designed a species-specific assay for was the honey mesquite tree. It is not wind pollinated, it's insect pollinated. So in terms of its ecology and evolution, we don't expect that it would release as much DNA that is through evolutionary processes designed to be caught in the wind and end up in our traps. And so if we could collect honey mesquite DNA, we would be pretty confident that this tool might be more useful than just a limited number of species that interact with the wind in very specific ways. And the short answer here, the short result, is that we were able to detect both species. So in terms of plants, we could detect a wind-pollinated species, we could detect a non-wind-pollinated species, a insect-pollinated species. So we started to think this seems like a useful method for studying terrestrial plants. So the next step we got to do is we decided to sequence our samples and not just take a species-specific approach, but now we're asking what plant DNA was in our samples. And we paired this with one of those more laborious traditional surveys. We walked transects across the landscape and visually identified all the plants that were around our traps. I also should have mentioned that this property that we were working on is a valuable teaching resource for our undergraduate program. And so every semester, teams of undergraduates go out onto this property and learn how to do plant surveys themselves and identify plants. And so we've got a very strong record, a very comprehensive history of all of the plant species that live on this property. And so the next couple of graphs I'm going to show you are diagrams that show how the number of species that we detected using airborne eDNA methods, the number of species that we detected only visually on our transects, and then how many species the two methods had in common. So we see right here, and I'm going to break this down by several different, not necessarily scientific, but intuitive plant types. And so here we're looking at plants that have showy flowers, bright yellows, bright whites. These are hard to miss on the landscape. And we see that we had a lot of species in common that we detected with both methods. There were some species that only eDNA collected, and there were also a decent number, actually more species, that visual surveys detected. And we attribute this to the fact that those showy flowers are hard to miss. Some of them are very small and might not be releasing as much DNA into the landscape or into the air as other species, but we see what we can conclude from this was for these showy, these visually obvious plant species, there still is some benefit. eDNA overall does a pretty good job of describing the system. It doesn't necessarily hit every single species, but it does capture some species that we missed on our visual transects. Now, when we look at these species that don't fall into the showy, the beautiful flowers section, these are grasses, forbs. To an aquatic ecologist, I would say that there's a little bit more boring, a little bit nondescript. Apologies to any of the plant experts in the room here. But this is where we really see airborne eDNA methods start to shine. We found almost all of these species using airborne eDNA methods. There were only 2 that we identified visually that we did not collect DNA for. And we attribute this again to the lack of those really pretty flowers, a little less eye-catching on the landscape. And this fits with that theme. Some species are hard to detect, and it looks like airborne eDNA is a useful tool for the toolbox that we can use to increase the amount of organisms that we detect in the environment. The last species category I'll call the trees. We found all 4 tree species that are known to occur on the landscape using eDNA, and we didn't find any using only visual surveys and not eDNA surveys. There were 2 tree species that we detected with eDNA only, but not in our visual surveys. Well, you might ask, well, how could you miss a tree, right? That makes the survey a little bit questionable. It's a big landscape, and these are tree species that we are aware of. We know that occur on the landscape, but they didn't occur on our random transects on that landscape. And so this is yes, it's an artifact of our sampling design, but imagine going out to a new forest or a new landscape where you don't know where what all the species are. You may not know what those artifacts, how to set out your visual surveys. And so this shows that you might miss some species, even some really obvious ones. And so, again, really encouraging that our airborne methods performed really well with this group of trees. So at the end of this group of studies, Mark got his master's, and he had me thoroughly convinced that this airborne eDNA method was going to be potentially really useful for surveying plants. I'd like to move on now to, I said I was going to start by saying, can we do it? Now we're going to look at, well, okay, if we can detect airborne eDNA from plants, can we use it to answer ecological questions? Are we just stamp collecting, or is it possible that we could learn something about the way species work out on the landscape? And so we're going to work on that same landscape, that experimental property, only this time we paired our sampling with one of our undergraduate clubs was doing a honey mesquite restoration project. They were actually, that honey mesquite tree is really thorny and is, depending on who you talk to, somewhat of an invader. And so there's interest in controlling it. And so they did this effort where they went out in a small portion of the property and they chopped a bunch of these trees down. And they actually did this twice over a couple-month span where they chopped trees down and applied some herbicides. And we collected airborne eDNA around the time that they were doing their sampling, here in the north, here in the south, and to the southeast. I'll point out that in this part of the landscape, the wind primarily blows from the northwest to the southeast. And I'm going to orient you to our sampling, and this is how our graphs are going to be laid out as well. So we collected an eDNA sample, those airborne eDNA samples from those dust traps at 2 points before that undergraduate club went out there with a bunch of chainsaws and started chopping trees down. They did that, it's represented on this is a timeline, so they did that at this first blue point. We took an environmental DNA sample in the middle. They did a second round of chainsaws and herbicides, and then we collected one more DNA sample before the year was over. Now I'll put the graph up here. Here we are quantifying honey mesquite DNA. This was the target of the restoration activity. The amount of DNA is on the x-axis here. This is the total amount of DNA. So here low numbers mean less DNA, high numbers mean more DNA. And then these blocks correspond with our sample collection times. So we sampled twice before the restoration activity. Undergraduates went out at this blue line with a chainsaw and chopped everything up. We sampled again. Undergraduates went out again with chainsaws and chopped down a bunch of trees, and we sampled again. And what I want to point out is that we saw low levels of airborne honey mesquite eDNA prior to this restoration activity. But then you send a bunch of undergraduates out with chainsaws and say, chop up that species and haul it away, and we see a plume of eDNA, especially right at the site that's closest to where they were doing all their activity. So this disturbance on the landscape, and it happened not once but twice here, this disturbance on the landscape seems to have resulted in a release of copious amounts of DNA into the air. So we interpret this to say, well, this airborne eDNA signal can give us a little bit of information about activity, things that are happening on the landscape and changes. As long as we had these samples, we also looked to quantify the amount of blue grama, or that grass DNA, in our samples. An important thing to note here, oh, and I should have said, 1110, either Americans or Europeans, I'm not going to say which, do our dates backwards. And so this is November 10th, November 17th. So blue grama was not the target of this restoration activity, but it's very common on the landscape. And you can imagine a bunch of undergraduates with chainsaws are stomping around and stirring up a lot of material. Even though in November, this grass species has reached the end of its seasonal activity. It is senescing. All of the biological activity is underground. The stuff above ground is dead and not particularly active, right? And we see this, we think we see this, from time 1 to a week later. You don't have to squint too hard to see a general decrease in the amount of DNA that's available in the environment. When our undergraduates stomp through, we see somewhat of a plume of DNA being released. We got to squint a little harder to tell this story, but we see DNA decreasing, and then this activity on the landscape, undergraduates moving through, we see a bunch of DNA being stirred up, and then blue grama finally is senesced away, and there's just no material to shed. But we see, so what we see in this study, what we think we saw in this study, is that we have we've captured seasonal activity and changes and events on the landscape with airborne eDNA. So at this point, we're really excited and convinced that airborne eDNA represents a pretty viable tool to learn about at least plants on the landscape. So now I'm going to shift, and in the last couple of minutes here, I'll talk about whether the sky's the limit. Is this just a plant thing? Is this just a neat tool that we can use to study plant communities? Or maybe can we learn some other things using airborne eDNA? And so about the same time that we were doing our work with plant DNA, several labs here in Europe were looking at using airborne eDNA methods to study animals. And so we wondered, I wonder if there was any animal DNA in our traps. That's one of the beauties of environmental DNA sampling. If you're not yet an eDNA sampler, you can use the same sample and look for multiple targets, multiple species in your samples. And so Mark had all these samples that he had collected and looked for plant species. We wondered, well, I wonder if there's any animal DNA in our samples. So again, I think this is the third time now I've shown you this slide just to remind you how we collected DNA and where we collected it. We used the exact same samples, and we used a universal vertebrate primer to sequence base and ask what animals were in our samples. There's a lot of information here, but I'll just point out Mark collected these samples approximately every 2 weeks. Again, starting in June and going to November, January, and then to the next June. He had the nerve to take his Christmas holiday off. I don't know why we missed some samples there in December. But in any case, lots of different animal species DNA were in our samples, including several different rabbit species that are very common on our landscape. If we hadn't detected those, we would have felt pretty nervous, like this isn't a particularly effective tool, because there's so many rabbits. But we got rabbit DNA particularly fairly consistently across the whole year. And then there were some species. This was my home city of Lubbock is in a particularly arid part of the state, part of the country. We don't get a lot of rain, and there really was one major rain event where we saw water accumulation on the ground. And after that one rainstorm, there's a species called the Texas toad, whose DNA appeared in one of our traps that was just positioned on the Playa Lake. There's a depositional wetland that fills up occasionally when we get a nice, healthy rainstorm. And so it appears that toad spends its time buried down in the mud, gets this big rain cue, says, okay, it's time to come out and reproduce and do my biological thing, and we happen to collect some of its DNA at the same time. If you look down at some of the bird species, we see some things like domestic chickens, which aren't on the property but are in the neighborhoods around the property, that we collect fairly consistently throughout the year. Other species we saw pass through that aligned pretty well with migrations across the landscape. And so we were really encouraged by this. Animals, we seem to reflect some seasonality, some behavior, and most importantly, we've got animal DNA in our samples. We tried to relate the amount of DNA or the number of species. This is alpha diversity, so the number of species we detected in any one sampling event to the amount of rainfall that we had received in the weeks prior to that event and the average wind speed prior to that event. And it does appear that the rain had some kind of effect, like perhaps it washed DNA out of the air, and then vice versa. The more wind you had, it seemed like the more material was being stirred up. And so some ecological things that we need to learn more about to really make this airborne environmental DNA tool useful. Switching gears a little bit, we were wondering, all of our sampling is at ground level, but we wondered if we sampled higher up in the air, would we detect eDNA differently? The Texas Tech National Wind Institute has this 200-meter environmental tower, weather tower. And so we were able to deploy, there's our dust trap again, we were able to position it at ground level and then at positions up all the way to the very top of this tower. So we collected airborne eDNA and we asked how much and what species did we detect over time. I will point out that we had this brilliant idea in early 2020 and we were going to sample at fairly regular intervals, but then the COVID pandemic happened and so our sampling scheme looks very unprofessional, right? Like who would design this sampling scheme? We sampled after 2 weeks, and then 140 days later, another 58 days. So the sampling is a mess. And just to head off some of the questions, we don't know right now anything about eDNA, airborne eDNA degradation, for example, in our traps. Probably some of the stuff that we collected on day 1 was no longer able to be amplified 140 days later, but we did the best that we could. At the end of this experiment, we sequenced DNA, we sequenced plant and fungal DNA, we sequenced invertebrate DNA, and vertebrate DNA using a couple of different markers, and this is not the best way to present this information, but we've got lots of diversity in our airborne eDNA samples overall. This is perhaps a little more interesting, but then a little busier. I point out, again, now we're looking at biodiversity. So this is alpha diversity, the number of species we detected across our different sampling periods. Each colored bar represents a different height. And we're still trying to understand these and figure out what's the absolute best story that we can tell, what's the most compelling story we can tell here. But I'll show you a couple of highlights just to show you some of the things we're thinking about this data set. First, we saw that the biodiversity, the types of species that we detected, changed over time. And I've got just a couple of species highlighted for you. For example, here's a beetle that is an agricultural pest. It emerges and is active in the early spring when crops are active at the same time, and that happens to be when we found that species' DNA in traps at various heights. This is also an agricultural pest. It's a weed, and it grows more towards the fall, and indeed, that's when we found its DNA to be most common in our samples. Here's an animal, the South Plains wood rat, that is a fall breeder, and the only time that we collected its DNA was in that fall period. So again, we've got this temporal signature in our airborne eDNA samples. Switch to a couple of other case studies, a couple of other highlights to show you. We've got this parallel harp ground beetle. The punchline here at the top is that we saw different species at different DNA heights. And so, for example, the ground beetle, we found it different times, but we only found it in our lowest traps, the ground level 1 meter off the ground and 9 meters off the ground. We never found its DNA really high. Giant ragweed, that's one of those allergens, releases copious amounts of pollen into the environment. Anyone that suffers from allergies understands that, and its DNA was everywhere. Great Plains toad, a slightly different frog species. Curiously, we found its DNA only at our 45, the wrong bars is highlighted here. These are supposed to be on these pink bars. We found its DNA only at 45 meters in height. I don't know why that is, right? So here we've got a signal of DNA that corresponds pretty consistently with a certain height, but this frog, this toad, is not hanging out at 45 meters off the ground. So some mystery. Oftentimes things correspond with our expectations. Sometimes they're mysterious. That's part of what makes the science fun, right? And then the last thing I want to cover, thinking about whether the sky's the limit, try to bring this back to my roots. I said I'm an aquatic ecologist, a freshwater ecologist. And so just out of curiosity, I really wanted to know, I wonder if there's any aquatic DNA in our airborne samples. Wouldn't that be great if I could just put a nice bow on this and bring this all together? So I had an undergraduate research class that I convinced that this would be an interesting question to look at. And so much like Elizabeth Clare did one of the first animal airborne eDNA studies in a small lab that was dedicated to the study of naked mole rats. There's a small contained environment. If we're going to find that DNA, we're going to find it in this small contained environment. We went to a zebrafish lab, a toxicology lab that had this whole wall of zebrafish tanks, and we set up airborne eDNA samplers so we could look for zebrafish DNA. This is not the best of pictures, but we had a passive eDNA sampler. So this is a cake pan with a liter of water in the bottom of it. And we had an active sampler. So this is a liter of water in this bottle, and there's an aquarium pump. We ripped out all the filtration, and so this is just bubbling air through that water. And we deployed these for different periods of time. We deployed them for short periods of time, as short as 5 minutes, all the way up to an overnight deployment. And so we took those water samples to the lab, we filtered them, and then we used species-specific PCR to look for zebrafish DNA. And we found zebrafish DNA floating through, apparently floating through the air in this lab, right? So this is the amount of zebrafish DNA. Again, it's quantity, so no DNA down here and more DNA up here. We found more eDNA in our active sampling than we did in our passive sampling. This is actually slightly above zero. It just looks very close to zero. We found a little bit of zebrafish DNA in our passive sampling. We didn't, however, see a time effect. There wasn't any. Our sample sizes were incredibly limited. I think we had 5 stations set up. And so we don't have any statistical evidence that the time that we spent collecting DNA affected how much DNA we collected, although I'm not convinced that that is a meaningful result just yet. I am, however, really excited that it seems like we can collect airborne eDNA of aquatic species. Why am I excited about that? Is that just a curiosity? Why not just go and take the water sample, right, as long as we're collecting aquatic organism eDNA? I think there might be a couple of cases where this might be useful. Some environments, some aquatic environments, are hazardous to sample, right? Climbing down these rocks and trying to collect aquatic samples down here would not be particularly, it's a job for a graduate student, right? Surely not a PI, first of all. And even a graduate student I wouldn't send down there, right? And so there might be better ways. Airborne eDNA might be an opportunity. That's me standing right there holding out a bottle trying to collect some airborne eDNA, right? Some other places can be difficult to sample, and I've been involved with some teams. We're looking at sampling DNA using autonomous vehicles, drones or little vehicles. A drone going out across a water body could offer us a different way to sample that water body. And so maybe there's some use for airborne eDNA sampling of aquatic organisms here. And then taking this, this is a little bit of a pipe dream, but taking this to its extent, right? If indeed there's airborne eDNA of all species floating around in the air, we could imagine someday that airborne eDNA gives us some opportunities for what I would say are extremely remote sampling of species. Could we, over here, and I apologize for leaving Spain off my map here, but could we be in Spain and collect airborne eDNA that would give us clues from the southern United States, northern Mexico, and the Caribbean, just because of the way that the winds are blowing. And so just to finish up here, I'll say that I really do think that the sky's the limit. I think, hopefully, I've shown you a lot of exciting things about airborne eDNA and its potential. We have a lot to learn in terms of our foundational understanding of environmental DNA. What is it? How does it accumulate? How does it degrade? What affects what's out there to be collected? We've got a lot to learn about what types of applications, airborne environmental DNA or old-fashioned eDNA for that matter, what we can learn using those samples, and indeed, how can we take what we've learned from our eDNA samples and connect it to those ecological questions, understanding the world around us, which I know is what truly motivates us all as scientists. So, with that, I will say gracias, muchas gracias for your attention, and I think I've left a couple of minutes for questions.
Any questions? Yeah.
Hi, thanks for your talk. It was really interesting. I was thinking about your airborne experiment with the tower, and the beetle stuck out to me in particular because I feel like there's so much difference in like the release of eDNA in the environment based on like hard bodies and soft bodies, that kind of thing. I was wondering if you looked at that at all along like the test gradient or anything like that.
Oh, this is a great question. So we have not looked at the hard body, soft body question I'll repeat, oh no, you had a microphone. I was gonna say I could repeat the question for online. But yeah, no, it's a good question, right? We know in aquatic, I'll tie this to my aquatic systems first and say that we know that some species, mushy species, slimy fish, are easier to detect in the aquatic environment, whereas if we look for crayfish, for example, they're very difficult to detect, except they molt and shed their exoskeleton as part of their growth, and we've found, other people have published, we've also seen this in our studies, that right after that molting period, they're mushy again, and they release a plume of DNA. So, tying this to our airborne, we have not looked at the species traits like that, but that's a really good suggestion. And so so for the beetle, we just found it at ground level, or no, you're talking about the one that, the agricultural pest that we just saw at a certain time.
I was talking about the beetle, like the slide with the beetle and the toad on the same page.
Mm-hmm, yeah. So in any case, yes, I think that's a great question, and I'll let you know the next time we come back. Next time I'm here to collaborate, I'll tell you what we'll look at that and see.
Cool. Thank you.
Thank you. It was really a great talk. I know you mentioned that you don't know much about degradation of the DNA, but when you saw the animal detection of DNA, you saw that the toad appeared at some point and then disappeared in the next sample that was like around 20 days after, or 20 days. Do you know if the degradation, or have any clue, is different for different organisms, or no idea?
We know nothing in aquatic systems or in airborne systems. We do know in aquatic systems, for example, several degradation studies have been done with several different species of fish. And it looks like DNA degradation rates are consistent across different taxa. Environmental conditions, however, abiotic conditions, influence degradation a lot, right? Hotter temperatures, more ultraviolet. Ultraviolet radiation is sort of up for debate, but higher temperatures, pHs, in aquatic systems, certain other items in the environment like dissolved clays and silts chemically react to DNA. They bind the DNA and protect it from degradation. And so I suspect more so, although I can't say this definitively, I suspect more so than the taxonomic differences, it's the environmental conditions would have an influence on our eDNA degradation. Mm-hmm.
Thank you very much. In your sampling design, your degradation will likely happen after you are captured the particle, right? So it's sampling for many, many days. So,
sure.
But it seems like it's in the dark, so you won't have much of UV degradation of the DNA?
Yeah, so it is in the dark, so ultraviolet light is not going to play a factor. It is, these traps are metal, and it is, much like Sevilla, it is very sunny and quite warm. And I imagine that those traps heat up considerably. And so I do think that there's probably considerable degradation going on. We just haven't quantified that. And any sample that we've collected with any regularity, we sampled after 2 weeks, and we haven't done smaller time frames. And one of the tricks, too, with the airborne eDNA out in the environment is our source of DNA is uncontrolled, right? We know it's from the surrounding landscape, but possibly beyond. And so we don't have a good handle yet of even what goes into our trap, and so we just haven't had the right setup to quantify degradation yet. I suspect laboratory setting where we have some animals in the room, or plants, and then remove them from the room is the way to go there, and that's just not not an experiment that we've done yet.
And second question is if you have considered like point samples in which you go to the field and filter for, let's say, 2, 3, 8 hours and then extract those things. We have only done our airborne eDNA work on that rangeland property so far, and haven't taken it to the next step of applying it and looking for targets out in unknown environments. I know recently, probably about a year ago now, the X Prize concluded a competition where various teams went out to a rainforest in Singapore and were challenged to this one of these brilliant $1 million prizes. They challenged the teams to, we're going to drop you off at a rainforest, give you 24 hours, or I think it's 24 hours of sampling and 24 hours of analysis. Tell us about the biodiversity in that rainforest. And not surprisingly, many of the teams used environmental DNA, combinations of little rovers that would drive across the landscape and scoop up soil and water, as well as drones that flew through the canopy. And so we have seen some successes of application, of spot detection of species, although I don't have any data to talk about that point.
Hi, very interesting talk. Thank you very much. Mike, I have a curiosity and then a proper question, I would say. The first one is that you have a mesh on top of your device, right? Mm-hmm. What happens if it rains? Does it wash away the dust, or?
Good question. We didn't ever have enough rain where we felt like our trap was being washed out. The answer is probably that it gets washed out. So some additional, like Mark showed early on, our sampling methods really matter. Not particularly surprising, but yeah, I think we've got things to develop. I think the field has generally started to agree too that active sampling, filtering, using a vacuum pump or something like that to pull air in is the way to go over a passive collection method. But yeah, thinking about, we absolutely could have had some washing of our traps and don't have any way to say what the effect was.
Okay, and the question is, we know that aquatic eDNA, metabarcoding, is not really quantitative, and this has or main limitation. How far is it for you from being quantitative in airborne eDNA? I know, very far for all of us.
You know, I'm on video here, so I will say that I recognize that a lot of people, that there's a lot of concern, a lot of legitimate concern about primer bias and other things making sequencing not quantitative. I will say, on the other hand, eDNA still isn't really quantitative, right? We know that some species shed a lot of DNA. We know some species shed less. We know that behavior matters, size matters. There's all sorts of reasons why you get a certain number of nanograms of eDNA and you don't know, well, how much biomass does that mean? Well, it depends. eDNA is really good, though. Time after time after time, study after study has shown it's really good at qualitative, at comparative abundances. We got more DNA here and there's more biomass. We got less DNA here and there's less biomass. And so I suspect there's room for quantitative metabarcoding there. And I know from the papers that I've read and just the papers that I really enthusiastically watch for, this field of quantitative barcoding is getting really hot, is emerging. And so I think if you ask me that question in a year or two, hopefully I'll have some good data to show. But the answer is right. For political purposes, I'll say, you know, right now it's in its infancy, but I think there's a lot of promise there.
Hi, thanks a lot for the talk. I was, it's a bit following up on these questions from Moncho and Anna, and I was more interested in knowing a bit more about the spatial accuracy of the detectors, because I don't know if you're showing everything you collect with the 9 detectors and that's it. At what point the environmental DNA decay, so then you cannot really assign a certain species to a certain location.
Mm-hmm. Yeah, when we outline, so I've spent most of my career thinking about what I call the ecology of eDNA. DNA is shed from an organism. It hangs around the environment for a while. Sometimes we're lucky enough to scoop it up and try to learn something about that DNA or the organism that shed that DNA. But what happens in the middle there? DNA accumulates, it transports, it degrades, it changes in its structure, goes from large fragments to smaller fragments, for example. And all of that ecology really matters. And so your question about the transport of airborne eDNA is a really important one, and not one that we have found. Well, let's put it this way. In our plant survey across the landscape, we used a reference database that included plants that we knew were on our landscape from decades of sampling. I suspect that there were many plants in our samples that were not on that 60-hectare landscape. You know, like any city, surrounded by neighborhoods that have all sorts of exotic, non-native. I'm sure we collected Spanish plants, European plants, Asian plants in our collection as well that probably were not pollen that blew in from Spain. And so I feel like this has been my answer for every question, and I apologize about this. This is, but this is indicative of, I think, what is an exciting emerging field. This is a great question. It's one that we need to learn more about, and I hope that in the future I can come back and give you a really good answer.
I think we can take this outside and debate about the...
All right.
We can debate more about the exciting times that we have ahead, no, in terms of airborne DNA. So thanks for coming. Thanks for inspiring us.
Okay.
And we can talk more about this.
All right. We'll take it outside. I like it. Thank you all.